Not that exciting.
All right, let's. The goal today is to finish acid-base balance, and I have a worksheet for you guys. You probably already downloaded it, and some of you probably already did it or tried it. That's fine. Those who didn't, we will go through a lot of those problems today, and then move into muscles. That's uploaded. It's there. I also have updated muscle IOs. I got rid of a few of the IOs that were there, and I think I changed around one or two. But the list is in Canvas for you. So, acid-base balance. Basically, our body needs to be within a very tight range of pH. And I never have you guys know numbers. These numbers you have to know. 7.35 to 7.45. That's the normal range of pH for the body. What's completely neutral pH? 7. So if our body is 7.35 to 7.45, what does that mean about our body's pH relative to neutral? Or slightly basic. Why do you think that is? Acid's bad. Depending on what generation you come from. Yeah, acid's bad. And our body, as a result of metabolic processes, constantly produce acid all day long. So we need a buffer for that acid, and the buffer's bicarbonate. We'll get there. But because the bicarbonate is in a 20-to-1 ratio, do you guys remember this? CO2 plus H2O yields carbonic acid, which then dissociates into hydrogen and bicarbonate. Okay? This is the acid. This is the conjugate base. Our body has a 20-to-1 ratio of bicarbonate to its conjugate acid, which is why we're slightly basic. All right? Because we have to maintain a constant buffering capacity, and we'll prove that later on. I don't know what's going on with this thing. Anyway. All right. So, just a few reminder things. pH is basically a measure of the concentration of hydrogen ions. If we have low hydrogen, not as much hydrogen, that's going to give us a high pH, and that's basic. If we have a lot of hydrogen, we have a low pH, and that's acidic. All right? This is back to like freshman year chemistry in college. So remember that. A lot of people get mixed up with this high-low thing. So low hydrogen, high pH. High hydrogen, low pH. All right. Why we care about it is that our body, when it gets too acidic, we go into a condition called acidosis. All right. When I give this the advanced version of this lecture again in spring, there's some slight difference in definitions. There's an acidemia and there's an acidosis. We won't talk about that today, though. If our pH gets too alkaline or basic, that puts our body into an alkalosis. Why? And I know we've discussed this. Is it important that we don't get too alkaline or or acidic? What does that do? Denatures proteins, right? And if we start denaturing proteins, that's bad. All right? Important thing to remember. In order to make things more acidic, we either add more acid or we take away base. Right? Both of those things can make things more acidic. So if we have lots of acid, things are going to get acidic. That makes sense, right? But if we have less base, that can also make things more acidic. Because what's the definition of base? It sucks in hydrogen ions, right? So if we have less base, we're going to have more hydrogen ions. Same thing with alkalosis. If we have low acids, that can make things more basic. If we have high bases, that can also make things more basic. All right? And you're like, oh my God, stop. You're like beating a dead horse. I promise you, some of you will screw this up at some point. Okay? Keep in mind, there's two ways to do this. And once we get to the problems, you'll see exactly why I spent a lot of time saying this. All right. We already talked about that. If you release hydrogen, you're an acid. If you suck up hydrogen, you're a base. That's all this is saying. So just kind of a reminder slide. And strengths of acids and bases. In our body, our buffering systems deals with weak acids and weak bases. But things we take into our body and metabolic processes can produce strong acids. All right? And any kind of strong acid into our body is going to change the pH. All right? And we talked about what that does, denatures proteins. So what we need to do is have a very tight buffering system so that we don't get too acidic, especially too acidic. Acidic is more of a problem than being too basic is. So acidosis is more of a problem than alkalosis, generally speaking. So we need to regulate very tightly. All right? The first line of defense are buffer systems. All right? We've been talking about this. Bicarbonate is the most important. Right? We already said that exists in a 20-to-1 ratio with the carbonic acid. That's why our pH is slightly alkaline. And it's a result of this equation right here. All right. We also have phosphate and ammonia as buffers. Those are going to be important in buffering our urine. And we'll see how that works in a little bit. For example, if we're in an acidosis and we use up all our bicarbonate, we start to use phosphate and we start to use ammonia. And those are really important for actually helping us to produce or reabsorb bicarbonate ions. And then proteins are also buffers inside of our cells because we don't want our cells to get too acidic. That's like the end game, right? We don't want the cells to get acidic because that's where the proteins are going to denature. So proteins can also serve as a buffer. We're not going to talk about those at all during this lecture. Okay? Don't worry about this. Why is the buffering system important? All right, our normal hydrogen ion concentration is very, very small. That's not a percent, that's the actual number, 0.00004 millimoles per liter. Not just moles per liter, but millimoles. So if you have moles per liter, that's 3 extra zeros added to that. So very small amount. But we produce 60 to 80 millimoles per day, which is way more than that, right? That means we have to be able to buffer that acid. We're constantly producing as a result of metabolic processes. All right? So that's 47,500 times more normal than normal hydrogen ion concentration. But we don't go in acidosis, right? Because we're able to buffer that. Important to remember. Not those numbers, but why we are buffering. All right, so when we buffer, we don't actually get rid of the acid. What we do is we convert it to something that minimizes the change in pH. So instead of having acids that change our pH from a 7.35 to a 7.34, 7.36 to a 7.34, we may change it from a 7.36 to a 7.35, right? So we minimize the pH changes. That's what buffers do. They don't get rid of the acid, they minimize the pH change. All right, we already talked about this. Our first line of defense is chemical buffer systems. That does a really good job. But we also have physiological buffer systems, all right? And that's where it's going to become really important to understand what's going on. 2 mechanisms, respiratory which deals with volatile acids. Volatile means they can, it's gaseous. Really, that's what it boils down to. Gaseous, so we're dealing with CO2. And where do we get rid of CO2? I heard it. Lungs, right? We breathe it out. So CO2, we've already talked about this too, is a what? Acid. So if we breathe out CO2, we get rid of acid. Okay? The other mechanism is through the kidneys, renal. All right, we can get rid of acids that way as well, and bases for that matter. So those act as buffering systems also. With the respiratory system, we can either that gets rid of CO2. If we get rid of CO2, what's going to happen to our pH? It's going to go up. Okay? If we breathe hard and deep, pH is going to go up because we're getting rid of CO2. If we have slow, shallow breathing, we're going to hold on to CO2. What's that going to do to pH? Down. It's going to make it more acidic. So if we're too alkaline, slow, shallow breathing will fix it. If we're too acidic, hyperventilation will fix it. All right? That's a compensatory mechanism. Kidneys, the same thing. We can either get rid of hydrogen. We'll talk about how that happens, which will make things more basic. Or we can hold on to hydrogen and get rid of bicarbonate. That'll make things more acidic. All right, so that's our second line of defense. All right, so we already said this. Carbon dioxide is an acid. Always. The more carbon dioxide you have, the more acid you're going to have because of this equation. Okay? Increase CO2 equals increase hydrogen. All right? We don't consider the bicarbonate in here as adding more base because we already have that 20-to-1 ratio. Adding another bicarbonate to that's not going to make that big of a difference. So it's the acid that's produced that's important here. All right, we just talked about rate and depth of breathing, all right, and what that will do to CO2. So this is just a reminder of what we just said. All right, so we talked about the respiratory system first thing, right? And we talked about the respiratory centers that are the central chemoreceptors that are sensitive to CO2 and hydrogen. If we have an increase in CO2 or an increase in hydrogen, remember it stimulates the medulla, which then stimulates our respiratory muscles to increase rate and depth of breathing. We get rid of carbon dioxide. So it is literally a feedback loop. We have sensors for this, and we have a way to get rid of it. By doing that, CO2 and hydrogen decreases. Okay, that's for volatile acids. Many of the acids we produce are non-volatile, so non-CO2. And if it's non-CO2 or non-volatile, we can't get rid of it through the respiratory. So we have to have another way of getting rid of it. All right? So non-CO2 acids, those are what we call metabolic acids. All right, and that's important to remember now. Metabolic acids. And these are a number of places they can come from. Aerobic respiration of glucose in the mitochondria of all of our cells. Anaerobic respiration, fatty acid oxidation, processing of proteins. All of that produces acids, right? Non-CO2 acids. So we have to have a way of getting rid of those and buffering them and getting rid of those. So if we can't get rid of them through the lungs, what's the other place we can get rid of them? Pardon? Urine, kidneys. Okay? So, two places we're talking about, the lungs and the kidneys. All right, what do I want you to know from here? We filter. Again, you don't have to know the numbers. It's just going through and showing you how this works. We filter about 4,500 units of bicarbonate, right? Because when we filter, that's non-discriminate. Like it's water and everything dissolved in it. We secrete about the same amount of hydrogen, right? So if we're filtering here, we're secreting here in the tubules. All right? We reabsorb almost all of that bicarbonate. All right, so we reabsorb almost all of that bicarbonate. Why? Why do you think that is? If we filter all that bicarbonate, why do we reabsorb almost all of it? So we can use it. We have to keep that 20-to-1 ratio. So we reabsorb. Remember, reabsorption is our kidney's way of saying, hey, we need to hold on to this. So we reabsorb almost all of that bicarbonate. But we are capable of producing even more bicarbonate. Okay, so our kidneys filter, reabsorb, and produce bicarbonate. And get rid of very little of it. What we do get rid of is related to the buffering capacity of our body. Okay, getting rid of acids or getting rid of bicarbonate in an alkalosis situation. All right, so we hold on to a lot of that bicarbonate. That's what that bottom part says. All right. Now, since we get rid of acids, hydrogen ions, into the tubular fluid, which we see right here, we've seen this picture before, hydrogen ions could potentially damage the cells of the tubules, right? And if they do that, our kidneys aren't going to work like they should. So we have other buffer systems in our urine that buffer that acid to, one, help get rid of the hydrogen ions, and two, prevent damage to the renal tubules. Okay? And that's phosphate and ammonia. Phosphate and ammonia transport hydrogen ions out and protect the renal tubules. All right. So how does this work? It's a process. All right, I'm going to go through the thing. That looks like, oh my God, what the hell's going on? When we secrete a hydrogen ion, that's how we're getting rid of hydrogen, right? We secrete a hydrogen ion. It combines with a filtered bicarbonate. Remember, we said the bicarbonate's freely filtered. We're secreting hydrogen to get rid of it. They form carbonic acid, right? Our renal tubule cells are able to split that carbonic acid into CO2 and water. All right? The water goes out as urine. We had one filtered hydrogen. We added a secreted hydrogen. We're getting rid of two hydrogens. So we got rid of the hydrogen. Does that make sense? That's where the hydrogen went. It went into water. We got rid of it. CO2 can freely go back into the cell, make carbonic acid, which dissociates into bicarbonate. Bicarbonate can go back in and act as a buffer. So this is the linking of secreted hydrogen to the reabsorption of bicarbonate. Right? To put that in simple words, for every secreted hydrogen, we reabsorb one bicarbonate. For every secreted hydrogen, we reabsorb one bicarbonate. All right, so that's how we take this bicarbonate, use it to get rid of hydrogen, and add it back. And it's all related to that equation. Does that make sense? If it doesn't, speak now, because we're going to make this a little more complicated. Like, sweet.
What you said about when it goes back into the cell for 6 weeks.
Okay. So when you have this water that goes out with the urine, The CO2 that's produced, it's a gas that can go right through the cell membrane. So it can dissociate right back into the cell. And your cell is constantly combining water with CO2 to form carbonic acid, which then dissociates. As an acid, it's always going to dissociate into hydrogen ions and bicarbonate. Okay? And then go back in. All right? I think it'll make more sense once we get to the next part. So this is what's happening on a normal everyday basis, like our normal acid handling. All right? But what if we have too much acid? What if we have too much acid being produced? All right? And we overwhelm bicarbonate. Like we don't have any more bicarbonate to use. We have to buffer with something else. That's when the phosphate and the ammonia come in. Okay, that's when the phosphate and the ammonia come in. And we'll talk about what they do in a second. Okay? In talking about these, this is the kidney version of when we get rid of CO2. When we breathe slow and shallow, we hold on to CO2. This is the kidney version of that. All right? In an acidosis, we increase our secreted hydrogens. We showed that. In acidosis, we secrete more hydrogens, which then goes through this process that we just went through. And we therefore increase bicarbonate reabsorption. Okay, during an acidosis, we increase hydrogen secretion, we increase bicarbonate reabsorption. And what we're going to talk about next is we also produce new bicarbonate if the bicarbonate is being all used up. All right, that means we're having so much acid, we overwhelm the bicarbonate buffering system. All right, this is going to be an important slide to go back to. If you're like, all right, how's the kidney going to compensate for an acidosis? How's the kidney going to compensate for an alkalosis? If we have an alkalosis, we decrease the amount of hydrogen we're secreting. We're not secreting it into the tubule. We don't want to get rid of hydrogen because we're too basic. All right? If we're too basic, that means we're going to decrease bicarbonate reabsorption. Like, if we're too basic, we don't want to reabsorb more. So we'll just get rid of it. It'll go out in the urine. Hence, we have a loss of bicarbonate in the urine. So, let me ask you something. In an acidosis, if Kelsey here had an acidosis and I said, pee in a cup, what would her urine be like? As far as pH goes. She'd have a low pH. It'd be acidic. If Kelly had an alkalosis and I said, pee in a cup, what's her pH going to be of her urine? Alkaline. All right. So the pH of your urine is going to reflect the condition that's in your body. All right. This is proof of concept. We're in an acidosis. We've used up all of our bicarbonate. So we're going to filter with either phosphate or ammonia. All right? So, same rules apply. We secrete a hydrogen, but we don't have any more bicarbonate. So that hydrogen is going to join with a phosphate. All right? Notice this hydrogen combined with this phosphate from one H. Now you have two H's. So you've effectively gotten rid of that hydrogen. What's missing right here? Where this red X is. You don't have CO2 going back in. Okay, you don't have CO2 going back in. So remember that CO2 was important for the regeneration of bicarbonate. So we don't have the regeneration. We don't have reabsorption. But you still have this process going on. So what you're doing is you're putting new bicarbonates in. Right? We don't have any more bicarbonate here. It's all used up. So we're not transferring it from here to here and reabsorbing it. So anything new that gets added, any bicarbonate that gets added to the blood is new bicarbonate, not a reabsorbed bicarbonate. The reason why? We've buffered by something other than bicarbonate. All right? So a new bicarbonate, net gain of bicarbonate. That's what we end up with. This is the same thing, only instead of phosphate, secrete hydrogen, adds to ammonia. You've gotten rid of that hydrogen, forming new bicarbonate. Right? If you look, we'll go back and forth between those two slides. They're showing the exact same thing, but just replace the phosphate with an ammonia. All right, so when we talk about reabsorption of bicarbonate versus production of bicarbonate, if you filter acids in the kidneys with bicarbonate, you reabsorb bicarbonate. If you filter acids in the kidneys with phosphate or ammonia, you produce more bicarbonate. To add to that buffering system. Okay, so this is just a summary of what we've talked about. And again, this is occurring in acidosis. This is occurring in acidosis, where the amount of hydrogen secreted by the kidney is way more than the amount of bicarbonate that's filtered. So we're putting new bicarbonates in. I'm not going to go through this, more numbers. This is just proving what I just said. More. Oops. Okay. So that brings us to this. All right, what are the numbers I already had you remember? 7.35, 7.45, which represents what? Normal pH range. All right. Anything below that is an acidosis. Anything above that is an alkalosis. All right. So if you look at a patient's chart and you see a pH of their blood, which is what you're looking at, or plasma, or arterial, the pH is going to tell you whether it's an acidosis or an alkalosis. But now we have to determine where the cause of that alkalosis or acidosis is. So we have two choices. It's either metabolic or respiratory. Okay? Two choices, metabolic or respiratory. If it's respiratory, where's the problem occurring? In your lungs. Anywhere else? No. That's the only place it can occur. So if we see a change in pH and we see a change in carbon dioxide that's corresponding to that pH change, we know we have a respiratory acidosis. What do I mean by that? Let's say we have a pH of 7.30. What would you say that is? Acidic. All right? And what if we have a PCO2 that's increased? Can an increased CO2 cause acidic conditions? Yes, because CO2 is an acid. All right, so respiratory acidosis. The metabolic pathway is a little bit different. All right, if we see a pH, we know the acidic or basic condition. But then if we see bicarbonate in our patient's chart, the bicarbonate tells us, and that's changing in relation with the pH, that tells us it's a metabolic problem. All All right, so if CO2 is driving the pH change, it's a respiratory problem. If bicarbonate's driving the pH change, it's a metabolic problem. Now, where can metabolic problems come from? Kidneys? Everywhere. Really, everywhere but the lungs. All right? So, that is, I'm glad you said kidneys, because we don't want to just think that the kidneys are causing metabolic problems. It could come from anywhere. We've already shown a slide that says that our body produces all this acid during the day. Okay, so metabolic could come from anywhere. So, if we see a pH of 7.30, we know that's acidic. We know nothing about CO2, but we see a decrease in bicarbonate. Can a decrease in bicarbonate make things more acidic? Yes. That's why I went through that whole thing where I said, remember, acid is an increase in acid or a decrease in or base, right? And that's what we're seeing here. All right, alkalosis, same thing. We can have metabolic or respiratory. Let's give a pH of 7.5. We'll just keep it simple. 7.5. If we see an increase in bicarbonate with that 7.5, can an increase in bicarbonate lead to an alkalosis? Yes. So therefore, since it's bicarbonate causing that problem or leading to that pH change, it's a metabolic alkalosis. If I see a CO2 that's decreased and a pH of 7.5, can a decrease in acid lead to an increase in pH or an alkalosis? Yes. So that would be a respiratory alkalosis. All right, we good to this point? All right. Because it gets more complicated. Because it's never just one variable. Okay? What this is showing you is a flow diagram of how you would analyze the problems we're going to go start talking about. So first you look at the pH. Is it low, below 7.35, or high, above 7.45? All the red I added because it said less than 7.4 or greater than 7.4. You have a range, not a single number. Okay? So it's below the range. All right? Two other sets of numbers I want you to remember. CO2, or PCO2, partial pressure of CO2. 22 to 26 millimeters of mercury. No, wait. I got that wrong. Sorry. $35 to $45. Millimeters of mercury. It's a partial pressure. Partial pressure is just a way of expressing concentration of gas in a fluid. Bicarbonate is 22 to 26 milliequivalents per liter. All right. CO2 is usually pretty stable across different panels. Bicarbonate, I've seen as low as 21 to 28 as well. All right. I don't care that you actually remember those specific numbers because it could be 21 to 28. It might change depending on where you're working. I want you to understand how to solve these problems. Okay? So these are the ranges we're working with. All right? So, we know that if we see a pH problem, let's say it's below 7.35, that tells us it's an acidosis. If we look at CO2 and it's above 45, that would be a respiratory. If we look at bicarbonate and it's below 22, that would mean it's metabolic. Right? We went through that. But your body is always, always, always. So we're going to do some practice problems, and the practice problems show that one of those, bicarbonate or CO2, is not changing. That's almost never going to happen. All right? Your body's always going to try to compensate. So If you have a metabolic acidosis, your respiratory system's going to try to compensate. Okay, so let's work through this. You have a 7.30. Tells us we have an acidosis. We have a 20 milliequivalents per liter of bicarbonate. That tells us we have a metabolic acidosis. All right. If our respiratory system's compensating, where are we going to see that change? CO2. We're going to see that change in CO2. And to compensate for an acidosis, what CO2 have to do? Do we have to keep CO2 or get rid of CO2? So you want to see CO2 decrease below 35. Okay? Does that make sense? Okay, and you can go through that for the rest of these as well. So if you get stuck, this is a good table to come back to try to figure out where you're going. All right? We'll get to the problems. All right, so a little more details about the metabolic and respiratory acidosis and alkalosis. This is the definition. What can cause a metabolic acidosis? So some of the things that can cause acetylsalicylic acid, aspirin, acid, increase hydrogen intake. Diabetes mellitus, ketoacidosis. That can cause a metabolic acidosis. Losing bicarbonate and diarrhea. So let's say you have a gastrointestinal infection and you are like diarrhea for 3 straight days. Remember, pancreas secretes a lot of bicarbonate to buffer what's coming from the stomach. And if all of that's going out in your stools, you're losing bicarbonate. What's going to happen to the pH of your body? It's going to plummet. All right? So bicarbonate loss through diarrhea can lead to a metabolic acidosis, as well as these other things. Okay? So, what can cause a metabolic acidosis? These are some of the things. What can cause a respiratory acidosis? So think about it. If we have a respiratory acidosis, that means our CO2 is increased, and the only way our CO2 is going to be increased is if we're not breathing it out. Okay? So brain damage, which could affect our pneumotaxic area. We're just not breathing. Pneumonia, emphysema, respiratory problems where we can't get rid of that CO2. Okay? Basically, any kind of lung disorder will cause a respiratory acidosis. All right, where we can't get rid of CO2. All right, what can cause a metabolic alkalosis? The exact opposite. Increased base intake. My brother, for example, used to just pop Tums, like a whole thing in a day. Okay? That's bicarbonate, sodium bicarbonate that he's taking in. You take in too much of that, you're raising the pH of your body. That's not good. Okay? So abusing antacids. It is a thing. You guys will see patients that do that. Vomiting gastric acid. So if you are like vomiting fairly frequently, getting rid of all your stomach acid on a repeated basis. Now this is assuming you vomit, that stomach acid builds back up again. You vomit, it builds back up again. You vomit. Okay, because if you vomit and don't let it build back up again, you actually start to pull stuff from your duodenum, and that could lead to a different problem. All right, we're not going to get into that. So that could lead to a metabolic alkalosis. Aldosterone excess, overuse of diuretics, things like that, getting rid of acids. Lead to a metabolic alkalosis. And respiratory alkalosis, anatomy midterms. Right? Anxiety. high altitudes, causing you to breathe too much. You're getting rid of that carbon dioxide. So that's going to put your pH up. All right, so be familiar with some of those causes. All right? Okay, we'll go through this systematically. I stole some of these slides. I get why they do it, but I don't know. So, what do we expect to see? You guys, God damn it. If you have a metabolic acidosis, so we already have defined the problem. What would you expect to see with pH? Metabolic acidosis, pH would be down. Specifically, give me a pH that would indicate a metabolic out. Yes, let's do it that way. 7.31. All right, that'll work. What would bicarbonate look like? 21. What would CO2 look like?
32.
Okay, 32. I don't remember what it says for compensation. Okay. So this would be the cause of our problem. This would be fixing. What would our respiratory system look like to give us that value of 32, a low CO2? Increase rate and depth of breathing. Okay? So if we have a patient that is in a metabolic, we have a diabetic patient that's in ketoacidosis, you would expect them to be going, not because they have a respiratory problem, but because the respiratory problem is trying to fix a metabolic problem. Does that make sense? Okay. Respiratory acidosis, give me a pH. 7.3. 7.3. Give me a bicarbonate. 20. Give me a CO2. 31. All right. So, respiratory acidosis, the boxes are what's causing the problem, right? Low bicarbonate is how we fix the problem. How are we going to get that low bicarbonate? All right. What do we have to do to lower that bicarbonate? To lower the acid? No. Yeah, this is fucked up. Why did I listen to you?
Sorry.
We have a low pH. To get a low pH, we need a high CO2. Oh, now it makes sense. Okay. Respiratory. All right. So what would we expect to see with bicarbonate? 27. Okay. So this is why you work your way through the problems. Like, see, I worked my way through the problem. And I'm like, something's messed up. This isn't sounding right. Okay. How do we get this bicarbonate to increase? We secrete H+. We reabsorb what? Bicarbonate. And we produce bicarbonate. Okay? So we secrete hydrogen ions. We buffer with bicarbonate. When that runs out, we buffer with ammonia and phosphate. And that's how we reabsorb the bicarbonate. That's why the bicarbonate goes up. Sorry about the... Sorry I listened to Caroline.
Yes. So if you check this person after they started compensating, you would expect to see high bicarbonate.
By the time you detect that they have a respiratory acidosis, they've already started to compensate. Well, that's partially true. Respiratory compensation. This will be important when we talk about this in a semester from now. Respiratory compensation is fast. Happens really quickly. Renal compensation takes a longer time. So over time, like if you measured like an hour, 5 hours a day, you would see that bicarbonate slowly start to creep up even more.
And if you didn't, then you know they have like major problems.
If you didn't, you would suspect they probably have renal problems too. Okay? I'm not going to go through the last two because I want to get to the problems, which is doing the same thing. Okay? I promise you. Okay, all of these showing the same thing. So let's do some problems. I have handouts for you. If you don't want to use the handouts, like if you need toilet paper, whatever. Mark, there will be one coming back from somewhere. If you guys have an extra one, send it back to Mark. All right. So here's how we do these. We'll go through one, and then we'll go through some of your practice problems. If I have a pH of 7.31, what does that tell me? I have an acidosis. Okay? Next, which one of these two is causing the problem? Pardon? Carbon dioxide. High CO2, high acid. Can that lead to an acidosis? Yes. Therefore, it's what kind of acidosis? Respiratory acidosis. All right. What's going on with bicarbonate? Pardon? Nothing. It's normal, right? So in this case, we would say there is no compensation. All right? I will never give you a problem like this on a test. It's too easy. Okay? If the kidneys were to compensate, what would we expect to see? Give me a number. Pardon? It's a lot of compensation. Usually if you compensate, it's not going to be a whole lot over what the range is. But yes, you would see bicarbonate increase. Why would you see bicarbonate increase? Secrete hydrogen, reabsorb bicarbonate, produce bicarbonate. Okay? So, what you want to do is identify your acid-base problem, identify the cause of the problem, identify the compensation, and explain why it's compensating. If you don't see a change, if you don't see compensation, explain what would happen to compensate. Okay? All right, so let's do problem.
Yeah, we just did one.
That was one? Let's do problem 3. 7.48, CO2 of 45, bicarbonate is 30. What's the problem here? What's the pH telling us? Alkalosis. That's spelled wrong. What's causing the problem? The bicarb. How do you know? It's high. So we would be a what? Metabolic alkalosis. Is there compensation occurring?
Yes. Oh, wait. No. Don't listen.
All right. Don't listen to Caroline. No, there's no compensation. How do we know? CO2 is within normal ranges. All right. Remember this. Within normal ranges, even if it's the high end of normal, it's still normal. You have to be out of the range. Okay? What would we expect to see happen to CO2 to compensate? CO2 would have to increase. Okay? What would our respiratory look like? Do what?
Decrease.
Slow, shallow breathing. So, you have the situation, your patient's going to be sitting in bed, and you're counting respirations, and you're barely able to see their chest move. Okay? Because it's going to be slow, shallow breathing. All right, how about 5? Tell me what's going on there. The pH tells us what? Alkalosis. What's causing the problem? So if you have a high CO2, that's going to make you more basic? No. So it's the bicarbonate. A key to these problems that works for me usually, I usually put arrows. pH is high. CO2 is high. Bicarbonate is high. Okay? That tells you which direction they're going. Right? All right, so more acid doesn't make things an alkalosis. More base does, though. So we have a metabolic alkalosis, but why is CO2 high? It's compensating. All right, so what does our breathing look like? Slow and shallow again. Right, slow, shallow breathing. That's going to help allow us to hold on to that CO2.
Will we ever have a case where, like, both of them would be contributing, the PCO2 and the bicarbonate?
I'm not going to give you one of those, but that can happen. That's what we call decompensation. That's the example that we gave earlier, where, like, if you went into renal failure and the values went down with the respiratory. Yeah, that would suck. No, I'm not going to give you those because this is a baseline. You have different types of respiratory acidosis and alkalosis. There's chronic and acute. And then you have anion gap and non-anion gap metabolic acidosis. We're not getting into that. That's for your Clin Med class. Okay? All right, pH of 7.3. This is what it is. That high, low, or normal? Low. Let's work it through it systematically. CO2, 50, high, low, or normal? High. Bicarbonate, high, low, or normal? High. All right, right away that tells you we have a acid-base problem. One thing's causing a problem, the other one's compensating. So right away, we haven't even done it. You know compensation is occurring. All right, so which one's causing the problem, which one's compensating? I heard. All right, we have an acidosis. That's clear. CO2 causing or bicarbonate's causing it? CO2. So it's a respiratory. All right. What's driving up that bicarbonate? How?
Secreting hydrogen and producing a reabsorbing bicarbonate.
Right. Secreting hydrogen, producing and reabsorbing bicarbonate. Now you can listen to Caroline.
Okay.
You can even sing songs about her.
Yeah, just like a few.
All right. How are we doing with this? Better? Okay. Let's add something else to the plate. All right. For those that you didn't hear, he called me a dick. pH, number 9. pH is what? It's normal. Carbon dioxide. Low. Bicarbonate. Also low. What the hell? How do we have a normal pH, but bicarbonate and CO2 are low? This is what we call complete compensation or full compensation. Okay, that pH is closer to what? An acidosis or an alkalosis? It's closer to an alkalosis. So we're recovering from an alkalosis. All right, which one of those values, when decreased, will lead to an alkalosis? CO2. So what kind of alkalosis did we have? Respiratory alkalosis. All right. What's compensating? Well, what's working on bicarbonate to compensate? What system? What organ? Renal or kidneys. All right. How are we decreasing the bicarbonate? We're excreting bicarbonate and holding on to hydrogen. Okay. So just the opposite of what we've been going over. We're holding on to hydrogen, getting rid of bicarbonate. All right, one more. Number 10. pH is normal. Carbon dioxide is low. Bicarbonate is low. All right. So, if you see the situation, both values are low but pH is normal, you still have an acid-base problem. Okay? So, what did we recover from? What are we recovering from? Acidosis. What type? Metabolic, because bases are decreasing. Why are acids decreasing? No? So, if we decrease bicarbonate, right, that's going to lead to acidic conditions. So we know bicarbonate's the cause of the problem, therefore it's metabolic. Why is CO2 decreasing? CO2 is an acid. Why is that decreasing? To compensate. So what is our breathing going to look like to compensate for a metabolic acidosis? Hyperventilation. All right, so the way we would write this is metabolic acidosis with complete or full respiratory compensation. Good? I'm going to do one more that's not on your list. All right. pH equals CO2 bicarbonate What's going on with this patient?
We're chilling. It's all normal.
Normal pH. Normal CO2, normal bicarbonate. What would you write? Yeah. You'd write normal. The point is, don't look for problems where the problems don't exist. Okay? If you see something like that, that is completely normal. I won't put something like that on a test, but everything's normal. Don't look for a problem where it's not there. All right, so we'll break there and come back with muscle physiology. All right, let's get started. Let's get finished. I'm done. I don't want to lecture. All right, skeletal muscles. All right, I super abbreviated your IOs, and I just want to. So if I skip over stuff and you're like, wait a minute, you need to go back, let me know, because I'm going to try to just skim over things and hit on things that are really important. So when we talk about skeletal muscle, realize that skeletal muscle, when you're dealing with it, is more than just the muscle. You also have connective tissue in there, which we saw in physiology, the fascia. You have nervous tissue to stimulate that muscle. You have blood to give nutrients to that muscle and take waste away, and things like that. So be aware of that. This is mostly anatomy stuff. All right, one thing about tendons, though. One thing about tendons is they are extensions of the muscle that attach muscle to bone. All right, if you didn't know this already, if you're attaching a bone to a bone, what's that called? Ligament. If you're attaching muscle to bone, that's a tendon. If you're attaching muscle to muscle, that's a, right, that's an aponeurosis. All right. The interesting thing about tendons is they are actually woven into the periosteum of bone. Like they're literally woven into the bone, like somebody stitched them in there. And they're part of the bone. That's why they are so tightly attached. It's not like they're glued to the surface. They are literally embedded within the matrix of the bone. All right. This is one of the important things you need to know. Ding, 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 ding. Within your tendons and within your muscles, you have two different types of receptors that will alter how your muscles contract. The first one is in a tendon. It's called the Golgi tendon organ. All right, yes, same Golgi that made name the Golgi apparatus in your cells. All right, Golgi tendon organ. That is a spray of sensory nerve fibers in the tendons that when they're being stretched too far, so like let's say, like, all right, I'm going to work out today, and I'm going to curl 200 pounds with one arm. Basically, what's going to happen, you're going to put such a strain on these tendons that your muscle is going to get the signal to relax, and you're going to drop it. Okay? So it's a way of preventing your muscles from picking up a load that it can't handle. And if you know somebody who can curl 200 pounds, that's impressive. With one arm. I can barely lift 10. All right. So what they contribute to an inhibitory pathway. All right. So they detect too much stretch. You don't want to stretch, or you're going to either pull the tendon off the bone or rip the muscle. So it's going to tell the muscle to relax, and you're going to basically drop the weight. The other reflex are these muscle spindle fibers. All right. within the muscle. So there are these smaller fibers within the muscle that have these sensory receptors on them called muscle spindles. And when they detect stretch, they will actually result in the contraction of the muscle to prevent overstretching. Okay, so they will contract the muscle to prevent overstretching. So the Golgi tendon organ detects tension, relaxes the muscle. The muscle spindle detects stretch and contracts the muscle to prevent overstretching. All right, they seem like the same thing. They sound like it, but they're a little different. Okay, one's relaxation, one's contraction. All right, both are preventing damage to muscle. Okay, we'll talk more about those when we get to the nervous system, the motor nervous system, towards the end of the semester. All right, going into the actual muscle. I'm not going to go through all of this, but if you take a muscle, it's divided into bundles or fascicles. Each one of those fascicles are divided into fibers, and each one of those fibers have a whole slew of contractile proteins that are organized into things called sarcomeres. All right, sarcomeres. So what you have at this level are a bunch of sarcomeres that goes from here to here, and they are arranged end to end to end all the way down. And they're made up of our contractile proteins, myosin and actin. All right, so what you see here in this picture is in the center of these is myosin. And, of course, myosin has these globular heads that are reaching up towards the actin, all right, on both sides. And then the actin has binding sites for the myosin so that myosin can grab on and pull the actin, right? And it pulls the actin toward each other and shortens the sarcomere. All right, we'll talk about how that happens. So add, and then you pull this way, shortening the sarcomere, right? If you have 100,000 sarcomeres end to end, one sarcomere may shorten just a little bit, but if you have them all end to end, that's a large shortening, okay, of the whole muscle. And that's the basis for contraction. All right, so how do we relate these proteins to what we see in a microscope? This, from here to here, is your sarcomere, from Z line to Z line. All right? This white part is actin only. These really dark parts are actin and myosin overlapping, so what we see right here. And this middle pink part is just myosin. Okay? Just to kind of show you how the proteins actually relate to what we see physically. And then this dark band, light band that you see under a regular light microscope makes these alternating striations, which is how we identify skeletal muscle. Okay? So taking it from the tissue level down to the molecular level, what are we actually seeing? All right, so just to kind of complete that discussion, when we look at this structure right here, put it into this cartoon so it's a little easier to look at. We have the I bands, which are your light bands. I bands, I for light. All right, again, those are just actin. The A bands are the dark bands. So I bands, A band, I band, A band. That's what makes up the striations, are alternating I bands and A bands. All right, then within the A band, the central part is called the H zone. And that's, again, just the myosin. So the I bands would be just actin. The H zone would be just myosin. The entire A band would be myosin and actin overlapping. All right? And dividing each sarcomere, which actually occurs in between, splits up an I band, is your Z line. All right, so actin, 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 actin. Let's do it this way. Myosin, myosin, myosin. Okay, that's what we're looking at. All right, surrounding that, we have this network of tubules called the sarcoplasmic reticulum. And the sarcoplasmic reticulum is a modified endoplasmic reticulum whose specific job is to, does anybody remember? Okay, back up. What does an endoplasmic reticulum do? Pardon? What did you say? Okay, make proteins. To make proteins. Okay, so endoplasmic reticulum either makes fats or makes proteins, depending on whether it's smooth or rough. All right, sarcoplasmic reticulum is modified so that it stores calcium. All right, it's a calcium storage unit. All right, so this is just kind of a summary of what all of that looks like. The actin, those are known as thick filaments, or thin filaments, actin, thin filaments. The myosin are thick filaments, if I didn't say that already. Okay, this is the contractile unit, okay, right here. That's one sarcomere. From Z to Z is your contractile unit. So, surrounding those contractile units are these sarcoplasmic reticulum. That's all this yellow stuff right here. And then they are subdivided into these transverse tubules. All right, and these transverse tubules are specialized structures that are attached to units within the sarcoplasmic reticulum that when a muscle is depolarized, it travels down the transverse tubule, opens up calcium channels in the sarcoplasmic reticulum, and then that calcium is released into the muscle. Okay, that's the point. And we'll talk about specifically how that happens in a minute. Right now it seems kind of nebulous. So we have these contractile proteins, then we have a sac of calcium surrounding them that can open and let calcium out. All right? We'll get there. It's important to remember that in order for a muscle to contract, you have to electrically stimulate it. And what electrically stimulates our muscles to contract? Action potentials from the nervous system. All right? So that happens at what we call the neuromuscular junction. All right? This is probably the most important part of this lecture, because any kind of muscle problems that you have are usually going to be at the neuromuscular junction. All right, so if you remember nothing from today, if I cover nothing else today, this is probably the most important. All right, also known, you'll see it as the NMJ, neuromuscular junction. All right, so what that is we have a motor neuron. The axon splits off, and where it's contacting the cell membrane of the muscle, which is called the sarcolemma, that's the neuromuscular junction. Now, each one of these points of contact right here is called the motor end plate. Right, the motor end plate, where you have an axon specifically going to a specific spot on the neuromuscular junction. The whole collection of them is the neuromuscular junction. All right. At this spot, you have a lot of nuclei, and you have a lot of, more importantly, mitochondria. All right, because in order to make a muscle contract, what else do you need besides electrical stimulation? Energy. All right, the neurotransmitter for all neuromuscular junctions is acetylcholine. ACH. All right, so what we're going to do is we're going to talk about what the stimulus is, how we get the action potential to release calcium, and how that results in a muscle contraction. So on the previous slide, we called that excitation-contraction coupling. All right, I like to draw this out. So pretend that's an axon, terminal. Okay? You have an action potential coming down. I'm skipping. Part of what I'm doing is not on this list. Okay? So you have an action potential. That's going to create a series of events that will take vesicles filled with acetylcholine, make them fuse with the presynaptic membrane, release that acetylcholine into the synaptic cleft. All right, remember that synaptic cleft is that little space between an axon and the cell membrane, whether it's a dendrite or whether it's a neuromuscular junction. All right, the acetylcholine is going to bind to acetylcholine receptors. All right, and acetylcholine receptors are not just receptors, but they're channels. Anybody remember what they're channels for? Nope. I'll give you a hint. It goes in. What did you say? Sodium. All right, so it opens up acetylcholine receptors, which is the sodium channel. These are what we call ligand-gated channels. All right, ligand-gated channels. So you open up sodium channels by binding acetylcholine. Sodium goes in. Anytime sodium goes in, what happens to membrane potential? Depolarization. You get more positive. Okay? So we start with the negative potential. It gets more positive. So that's going to that's going to depolarize the muscle cell membrane, and it's going to get to a point, right? If we remember this graph, you depolarize a little bit more, you reach threshold, and you send an action potential. Okay, so that's going to spread across the muscle fiber. All right, I like to draw this way. Let's use green. The green is my sarcoplasmic reticulum, okay, which contains a bunch of calcium. All right, dying to get out. All right, keep in mind that all of this is within the muscle cell. All of this is happening inside the muscle cell. All right, so when we depolarize the membrane, we're also going to depolarize these transverse tubules. Remember the sarcoplasmic reticulum, then we have these transverse tubules at regular intervals. You're going to depolarize these transverse tubules, and that's going to open voltage-gated calcium channels in the sarcoplasmic reticulum and allow calcium to come out. Okay, so the nerve released acetylcholine, opened up ligand-gated sodium channels. Sodium went into the muscle cell, depolarized it, causing an action potential that traveled down the transverse tubules, opened up voltage-gated calcium channels, and allowed calcium to come out. Okay, does that make sense? That's where we are. That takes us to the end of 4 here. All right, specifically the way this looks, so this is the same picture we drew. This is the axon terminal. The little blue is acetylcholine. In your muscle cell membrane, what you have here are your acetylcholine receptors up here towards the top, and your voltage-gated channels down here. Okay, so this will allow sodium in, which will go down and depolarize these voltage-gated sodium channels and start the action potential. Okay, so ligand-gated channel depolarizes you just enough to open up a lot of sodium channels down here. All right, so that's specifically how it's organized. All right, 2 differences here. When we are opening up acetylcholine receptors and allowing sodium to go in, which is taking our potential, minus 70, up to minus 60, minus 50, and let's say that's as far as it goes, and then we get another round of acetylcholine, which takes it up to maybe minus 40, and that's as far as it goes. That's what we call end plate potential. Okay, that's just the depolarization of the membrane. When we get to the point where we activate these voltage-gated channels, like let's say that happens at minus 35, which is not true, then that's when we trigger the action potential. All right, so end plate potential is how depolarized are we making the muscle cell membrane. The action potential is the actual signal where we signal these T tubules to open up calcium channels. We talked about all that. Okay. I went a little too far. This is just showing you end plate potential. This would be the action potential. In order to stop, okay, so question. When we stimulate a muscle cell with an action potential, do we always want it to remain active? No, so we have to stop it. All right? As long as there is, I'm not even going to bother drawing. As long as there is acetylcholine present, you're going to depolarize the muscle cell membrane, like that end plate potential. As long as acetylcholine is present, you're going to continuously depolarize the muscle cell membrane. You may not get an action potential, but you're going to continuously depolarize the membrane. All right. So in order to stop a contraction, to stop the depolarization of that membrane, we need to get rid of acetylcholine. Okay? So you have a built-in mechanism in this area, like between, within the synaptic cleft, to get rid of that acetylcholine. All right? It's an enzyme called ACHE, acetylcholinesterase. Okay, acetylcholinesterase. What acetylcholinesterase does is it takes acetylcholine, breaks it into an acetyl group and a choline. The choline is taken back up into the axon terminal where you can make acetylcholine again, and the acetate goes into something else. Okay, so acetylcholinesterase breaks up acetylcholine. Part of it's reabsorbed into the presynaptic terminal. The rest of it goes someplace else. And you shut down stimulation of the muscle. All right, this is really important to understand. All right, if we want to put it in terms of over here, as long as acetylcholine is binding to these receptors, we're opening up channels, depolarizing. If we have acetylcholinesterase present, we don't have acetylcholine, and no more nerve transmission. Okay, this is occurring in the synaptic cleft. So, if we have a patient who has a neuromuscular junction problem where they can't stimulate their muscle cells to contract effectively, maybe like myasthenia gravis, how could we help them? Guesses? I'll give you a hint. It has to do with acetylcholinesterase. We can inhibit acetylcholinesterase. We can give a drug that inhibits acetylcholinesterase. If we give a drug that inhibits acetylcholinesterase, what happens to acetylcholine in the synaptic cleft? It floods.
But then what happens when, like, they don't want to contract the muscle? If they constantly have that.
So, muscle contraction and the release of neurotransmitters is a, how do I put this? It's not an all or none process. So when you have an action potential go down, you're not releasing all your acetylcholine. It depends on how many action potentials you're sending down. So if you have a problem with not sending enough action potentials down and you're not releasing enough acetylcholine, then to help that, you flood the membrane with acetylcholine, and that's going to give them the same kind of contractions that you or I would have without a problem. All right, so they're able to clear that at a rate that's similar to ours. Okay, that's really important because a lot of disorders, like I said, occur at the neuromuscular junction and are related to how we can manipulate acetylcholinesterase. So make sure you understand that acetylcholinesterase problem and how this part works especially. Okay. Certain drugs we can give also that can moderate the neuromuscular junction. One class is inhibitors. So curare or curare-formed drugs. What they do is they block, specifically, I said they're acetylcholine receptors. They're nicotinic acetylcholine receptors. Remember with parasympathetic and sympathetic, we talked about nicotinic versus muscarinic. These are all nicotinic type. So what these curare-formed drugs do is they block nicotinic acetylcholine receptors and prevent acetylcholine from binding. So if acetylcholine doesn't bind, what happens? You don't open up sodium channels. And if you don't open up sodium channels, do you depolarize the membrane? No. So can your muscle contract? No. So instead of getting to action potential level, we keep our depolarizations really low. We never actually get the stimulation to contract. All right. Another inhibitor is botulinum toxin. Botulinum toxin. All right. What that does, it works a little bit differently. It decreases the release of acetylcholine from the nerve terminals. So instead of acting here, it's acting here. Right? It's preventing those vesicles from releasing acetylcholine from the presynaptic terminal. All right. If you have that drug, what happens is you never have acetylcholine being released. Therefore, you have nothing binding to the receptors. Therefore, you're not opening sodium channels. Okay. Different mechanisms. One blocks the receptors. One never allows the receptors to be filled at all. Okay. So the consequence of that is, again, low depolarization, you never reach threshold, you never send an action potential, you never have a muscular contraction. All right, stimulants. Acetylcholine-like drugs. We'll use nicotine. Why do they call it the nicotinic choline receptor? Nicotinic cholinergic receptor? Because it also binds nicotine. All right, so nicotine binds to the same spots on acetylcholine receptors that acetylcholine does and activates them. Okay? But nicotine is not destroyed by acetylcholinesterase. So if it's not destroyed by acetylcholinesterase, it doesn't come off. So you constantly have sodium channels open. So you have a prolonged stimulation of your muscle. Does that make sense? Okay. Another drug that can have this effect, stimulation effect. Now keep in mind, this is binding to the receptor, anti-acetylcholinesterase. You block acetylcholinesterase. This is kind of what we were talking about earlier. If you block acetylcholinesterase, you block the degradation of acetylcholine, so it's going to remain on the receptors, causing channels to be open and a prolonged stimulation effect. All right, if that happens too much and you don't never clear that out, okay, like you say there, nerve gas, right? What does nerve gas do to people? What can it do to people? Kill them. How would that kill somebody if you overactivate their acetylcholine receptors? Like they're contracting muscles, right? That's good, right? Pardon? You only have so much ATP you can produce. If you tire out your respiratory muscles, what's going to happen? not breathing anymore. Okay? So that's how these would work for, like, killing people. Let's think about that for a while. Because we all like to kill people, right? We all think about it. I just read all the Dexter books recently. Did you guys know that show was based on books? I read all of them. Way different than the show, but good books. All right. Last year when I taught this, it seems like everybody's always interested in these drugs that can kill you. So I like to go over them because they're interesting, and you guys have a lot of questions usually, most of which I can't answer, but we'll try. Sarin is another one. In fact, I have a whole case study on sarin. I'm not going to give it to you. I use it for undergrads. We don't have time for that. Nerve gas. Sarin is a nerve gas. Inhibits acetylcholinesterase, raises acetylcholine levels, and it's considered 26 times more deadly than cyanide. So, again, Allison, did you ask the question? What happens if you overstimulate? We're giving these patients doses, small enough doses, to where it's going to be therapeutic. If we were to give them a high dose of these things or something that acts more strongly, then it can produce these kind of effects. Okay. The thing about sarin is it forms a covalent bond on acetylcholinesterase. Right? Forms a covalent bond. What's the difference between a covalent bond and an ionic bond, for example? Pardon?
Covalent shares.
Yes. And it's stronger and more permanent. Right. So if you have a covalent bond on acetylcholinesterase that prevents it permanently from acting, you permanently deactivate it, what's that going to happen? Or what's going to happen? Right? Your muscles are going to fatigue. You're never going to shut off that response. So death occurs by, do you wonder what asphyxia is? Asphyxia. That's a typo. I didn't catch it in time. What's asphyxia? Not breathing, right? So how can we treat this? Atropine, cholinergic drugs. Right? We stimulate the cholinergic system and override that anticholinergic drugs, that prevent this from happening. Okay? Reactivates acetylcholinesterase. Therefore, we solve the problem. All right, so there are solutions to this, but all right. We're not going to get through even half of this lecture. That's fine. That's good. All right. Yeah. So we've got 12 minutes. I want to make sure that I cover one more thing, which happens here. No, it's not that far away. Okay. So all of that we talked about was occurring at the neuromuscular junction. I went a little bit farther and said, all right, when we have a depolarization of the muscle cell membrane, that causes a action potential to go down the transverse tubules, opening up calcium channels in the sarcoplasmic reticulum, and calcium comes out. That's what this is showing. Action potentials in the sarcolemma, travel down the transverse tubule, open up calcium channels in the sarcoplasmic reticulum, calcium comes out. All right? Why do we need calcium to come out? Actually, before that, this is kind of what that looks like. In the T tubules, this is depolarization where the inside gets positive now. That activates these DHP receptors, which the best analogy is like a spring, and it opens up these trap doors in the sarcoplasmic reticulum and allows calcium out. When we repolarize, that shuts that trap door. Calcium doesn't come out anymore. So Question for you, though. We need calcium to stimulate a contraction, and depolarizing the sarcoplasmic reticulum releases all our calcium. How do we get calcium back in there? Pumps, right? So we have these calcium pumps, bless you, that pump calcium back in. That is energy dependent. So a lot of energy with muscle contraction actually is spent on putting calcium back into SR. SR being sarcoplasmic reticulum. Okay. So, let's continue. We've released calcium. All right. Why is that important? All right. Calcium comes out. Calcium, if we look at actin here, actin are all these balls, these white balls right here. Okay. The binding site for myosin is covered by this molecule called tropomyosin. Right? So the binding sites for myosin on actin are covered by tropomyosin. And that tropomyosin is attached to a protein called troponin, which acts like a lever. So calcium comes out, binds to troponin, moves tropomyosin out of the way. This is the reason we have calcium. Binds to troponin, moves tropomyosin out of the way, and now you see all these purple dots here. The binding sites for myosin are unblocked. So calcium unblocks the myosin binding sites on actin. So calcium, another way to say that, calcium allows myosin and actin to bind to each other. Okay? Calcium allows myosin and actin to bind to each other. All right? Actin and myosin then link up to each other, and myosin will pull actin in one direction. Okay? So, calcium comes in, exposes binding sites on actin for myosin. Myosin binds to actin, and it pulls actin. I used to have markers that attach to each other. Pulls actin in one direction, right? If we're talking about the sarcomere, it's pulling actin toward each other, right? The contraction. All right. So the way this works is that, actually I like the other one better. Both of these are showing the same thing. When myosin and actin bind to each other, pretend this is myosin. Pretend the blue marker is actin. All right? It's cocked back, like it's ready to go this way. Stab. All right. ADP is bound, right? What is ADP? What's that a byproduct of? You broke down ATP, right? So the ATP broke down to cock this back, right? It's like a gun. You pull back that hammer. So ATP was used to cock that back. Myosin, actin bind. As soon as they bind, myosin goes, whoosh, moves actin forward one unit. ADP comes off. Okay, ADP comes off. Like pretend it happens so fast that ADP is like, whoa, it falls off. ATP binds, it releases. ATP is hydrolyzed, goes back, grabs another spot on actin. ADP, whoop, binds. ATP, hydrolyzed, and it goes over again. It throws markers. Oh. Okay. So that's how we get, if you see, I recommend going on YouTube. If I can find the one I like, I'll share it. Like, you see actin and myosin interacting, it's like, this is what they look like. Myosin's walking the actin in a certain direction. So we call it the walk-along theory. So all of this is showing you the same thing I just showed on the previous slides. This is just another way of looking at it. Okay, so that's how we get this contraction. Bring the actin close to each other. So now we have a contracted muscle. It relaxes. They go away from each other, right? So as long as you have acetylcholine stimulating the muscle cell, as long as you have calcium present, and as long as you have ATP, you're going to have muscle contractions. Acetylcholine, calcium, and ATP. All right, so the energy part of it. Excuse me. Where does the... Excuse me. Where does the energy for muscle contraction come from? All right. Your answer is ATP, right? That's where the energy for everything comes from. But we only have enough ATP in our muscle for 1 to 2 seconds of a contraction. That's any muscle, 1 to 2 seconds of a contraction. That's not very long. Do our contractions last longer than that? Yeah, especially when you're talking about muscles for posture. They're constantly contracted. All right? So how do we get enough ATP to sustain longer contractions? One of the places is creatine phosphate or creatine phosphate. All right? What that does is it's a molecule that basically holds an extra phosphate so that when you blow through that ATP, what's the byproduct of ATP? You break it down and you form ADP. So this creatine phosphate, which is like 5 times more abundant in your cell than ATP, will just donate its phosphate to ADP. So you automatically generate your ATP again, right? And if it's 5 times more abundant and your contraction with ATP alone lasts 1 second, then you're going to have a contraction that lasts about 5 seconds, right? So you can just increase your contraction from 1 second with ATP alone to 5 seconds with creatine helping you out. Does that make sense? Okay. So creatine phosphate is probably, I would say that's your first line to sustain contractions. After that, you need more energy for more contractions. Your second source would be glycolysis. All right, that's anaerobic metabolism, so you're not producing like a whole lot of ATP, but you're generating more, right? So creatine phosphate just allows you to regenerate the ATP you already have. Glycolysis allows you to produce more ATP, but not in very high abundance. All right, and glycolysis, remember, is occurring in the cell, not the mitochondria. So where does most of our energy come from? Okay, so that can allow us to get, since we're adding enough ATP to the system to get a sustained contraction for up to 1 minute. So now we've gone from like a 5-second contraction to 20 times more, right? 20 times, no, not 20 times more. What's... 20, 46, 20 times more. 20, 40, 60, right? No, 20 times 5. Oh, my God. 12. 12 times 5 is 60. So you take a 5-second contraction, 12 times more is 60 seconds. Jesus Christ. All right. So enough ATP to 12 times the amount contraction. That's okay, but we need even more than that. So that's where the citric acid cycle and electron transport chain, which produces about 36 to 38 ATP per glucose molecule, that's where we get most of our energy. So this is from our all of our activities that we do on a daily basis, like running, jumping, walking, other things. Okay, so those are energy sources. I think that's where I'm going to leave it. I'm not going to hold you responsible for the rest of it.
Wait, what? Really?
Yeah, I think so.
Like not recording
Hmm?
Like inactivated refrigerants?
No.